So today we will discuss about the TI plasmid. Okay, so before going into the TI plasmid, I would give the brief introduction about the Agrobacterium tumefaciens, which is the host of the TI plasmid. Okay, so Agrobacterium tumefaciens is a rod-shaped gram-negative soil bacterium that is naturally infected the dicot plants. Okay, not the monocot plant, but it is sometime it is useful for the monocot plants by using different modification. After that we can use this Agrobacterium tumefaciens as a vector system for transfer of any kind of DNA from means desired DNA to the means plant system. Okay, so the agrobacterium are mainly there is different strain of the agrobacterium, but the most common strain of the agrobacterium is agrobacterium tumefaciens and agrobacterium rhizogens. Okay, and which are essential to naturally infect the plants at the wound site and causing the crown gall as well as the hairy root disease. Okay, so what I wanted to say is this is the two most common strain of the agrobacterium species. One is the Agrobacterium tumefaciens and another one is the Agrobacterium rhizogens. Okay, this Agrobacterium tumefaciens is responsible for the crown gall disease. Okay, and this Agrobacterium rhizogens is responsible for hairy root or root mat disease. Okay, and that plasmids which it contains, the which is contained by the Agrobacterium tumefaciens is the TI plasmid. Okay, tumor inducing plasmid. Okay, and the Agrobacterium rhizogens is containing the RI plasmid, that is the root inducing plasmid. Okay, so these two type of plasmid is like other plasmid, how we use the plasmid to transfer the DNA from one cell to another cell. Like that, this plasmid is also helpful for transferring or act as a vector system. Okay, so in natural condition, this tumefaciens is responsible for the crown gall disease, and this rhizogens is responsible for the hairy root disease or the mat disease. Okay, and this tumefaciens is having the TI plasmid, and this rhizogens is having the RI plasmid. Okay, so TI plasmid is a tumor inducing plasmid, and it is a extra chromosomal genetic material which is found in the Agrobacterium tumefaciens, and it causes the crown gall disease in certain dicot plants species. Okay, and this TI plasmid can be transferred to the plant chromosome because it is having certain kind of sequences or certain kind of the genes which is responsible for the transfer of particular segment of DNA into the plant cell. Okay, and it also has the ability to transfer other foreign DNA sequences. If we will put these foreign DNA sequences into the plasmid, then it can also be transferred into the plant chromosome. Okay, and the size is around 200 kb. Okay, the TI plasmid is around the 200 kb large. Okay, so TI plasmid is means classified according to the opine producing plasmid. Okay, this opine is nothing but it is the nitrogen, or it is the energy source for the Agrobacterium tumefaciens bacteria. Okay, so it is based on the what kind of opines it is producing. This plasmid is classified. So suppose it is producing the nopaline opine, then it will the plasmid will called as a nopaline plasmid. If it is producing the octopine as a opine system, then it is called as the octopine plasmid. And if it is producing the agropine, then it is called as a agropine plasmid. Okay, so there is these three are the means very common type of opine synthesized TI plasmid. Apart from that, there are different varieties of TI plasmid also. Okay, so opine is their energy system, like nitrogen carbon source is called as a opine. Okay, so this is the structure of the TI plasmid. Here there is three major regions is there. One is the TI-DNA region, tDNA region. Okay, so this is the tDNA region. Here you can see this is the tDNA region. In the tDNA region, there is a left border sequence. Okay, and there is a auxin gene, there is a cytokinin gene, and there is a opine gene, and then after that there is a right border sequence. Okay, so when this plasmid is transferred from bacteria to plant cell as it is, then what will happen is this auxin cytokine gene will produce in more amount. Okay, so as you know that this auxin and cytokinin is responsible for the development of shoot and root generation. So overproduction of this gene will cause the crown gall disease in the plant. Okay, so and this opine will helpful for the metabolism of this TI plasmid or the agrobacterium tumefaciens multiplication. This opine will give the energy. Okay, so this is the tDNA region, which is very much essential of the TI plasmid. This tDNA region we can modify to transfer our target gene into the plant cell. Okay, so for inducing a tumor, only the tDNA is transferred for one cell to another. Okay, so if suppose only this part will be transferred, then it can cause the crown gall disease. Okay, and by modifying this particular plasmid into like for our beneficiary, we can transfer our target gene also to the plant cell to develop the modified plant. Okay, so the second region is the vir region. Okay, or we can say like this is the virulence region. Here this is the virulence region of the TI plasmid. Here it contained lots of virulence genes, which is responsible for the excision, integration and transfer of this tDNA into the plant chromosome. Okay, and the third one is the opine catabolism region. Here the opine catabolism region. So suppose the nopaline type of opine it is producing, then the catabolism will be happen by producing the opine catabolism protein. Okay, so this opine catabolism protein is required to catabolize the opine which is produced by this TI plasmid. Okay, so this region catabolize the opine, which are specialized amino acid, or we can say like it is the nitrogen carbon source. It is responsible for the catabolizing opine produced by tDNA region, and opine often act as a source of nitrogen in the bacteria. Okay, so this region only require when you are having the opine region in your tDNA. Okay, suppose you are not having the opine region, then no need to like also insert this opine catabolism. Okay, natural condition it is produced because the TI plasmid causing the infection inside the plant cell. Okay, so that's why to multiply the agrobacterium tumefaciens it requires energy. So that's why this opine will synthesize and metabolize this opine system. Opine catabolism is required. Okay, then apart from these three essential region, there are other essential region like the plant hormone synthesis region. Okay, so this region is responsible for the synthesis of essential plant hormone like the auxin and cytokinin. Okay, and the second one is the conjugation region. Okay, it is helpful to transfer the TI plasmid during the transfer of tDNA from one cell to another. Okay, and it is this tDNA border sequence. Here there is two border sequence. One is the left border sequence and this is the right border sequence. Okay, and this sequence is 24 base pair direct repeat. It marks the border of the tDNA, and it is important for the transfer of plant chromosome. Okay, so in the previous experiment it has seen that if the right border sequence is abolished from the TI plasmid, then the transfer process is also inhibited. The transfer of tDNA from one cell to another cell will be also inhibited because of the absence of this right border sequence. So it is very much essential to having the right border as well as the left border sequence in the tDNA region, so that it can efficiently transfer from one cell to another cell. Okay, so this is the structure of the TI plasmid, which is having three common regions. One is the tDNA region, which is the main, it harbors the target gene, okay, here. And the second one is the virulence gene, which is responsible for the excision, integration and transfer of this tDNA region from one cell to another cell. And third one is the opine catabolism, okay. Or if suppose what kind of opine it is producing, so to metabolize those opine, there is the opine metabolism region is there, okay. So now, we wanted this phenomena to, like in the RDT technology, okay. So how we will use this TI plasmid to transfer the vector, transfer our target gene from one cell to another cell, okay. So the desired DNA sequence can be inserted into the tDNA region, because it is having capacity to insert the tDNA sequence into the tDNA region of the TI plasmid, and then the TI plasmid contained in an Agrobacterium tumefaciens is used to deliver and insert the genes into the genome of the plant cell, okay. But to means transfer one cell to another cell, there is lots of limitation, like as it is using this TI plasmid, we can't transfer the our target gene from one cell to another cell, okay. So what are those limitations? Like the production of phytohormones by the transformed cell growing in culture prevent them from being regenerated into the mature plant. Therefore the auxin and cytokine gene must be removed from any TI plasmid derived cloning vector, right. So suppose if we will transfer this TI plasmid by harboring our target gene, so what it will happen is, when you transfer this TI plasmid from one cell to another cell, then what will happen? This auxin and cytokine gene will be also produced simultaneously, so it can cause the disease, the crown gall disease, into the another cell where it is going to transfer, right. So that's why this auxin and cytokine gene, when you are transferring your target gene from one cell to another cell, that time you have to remove these two genes by modifying this TI plasmid, okay. The next one is the gene encoding opine synthesis is not useful to a transgenic plant and may lower the final plant yield by diverting plant resources into the opine production. Therefore, the opine synthesis gene should be removed. Okay, because opine is required by the Agrobacterium tumefaciens, right? Not by the plant cell. So when you transfer your target gene with by means with this opine gene, then what will happen? The plant cell will also produce, means spend some energy to produce this opine gene, which is not useful for the plant cell. It is useful for the agrobacterium, right, bacteria. It is not useful for the plant cell. So that's why we don't want to spend much energy to synthesize this opine gene. So that's why we also have to remove this opine gene as well. Okay, with auxin and cytokinin, we have to remove the opine gene also. Then the third thing is TI plasmid is very large, like as we earlier mentioned, it is around 200 kb. So that's why this large plasmid is not preferable for vector purpose, right? So for using as a cloning vector, we have to remove the extra parts which is not essential for the transfer of this TI plasmid, this tDNS region, which is not essential. There is lots of non-essential parts is there, so that also we have to remove, because this the size is very large. Okay, then because the TI plasmid doesn't replicate in the E. coli, therefore it can't be cloned into the E. coli. Okay, so because it is having the origin of replication for the Agrobacterium tumefaciens. Okay, so that's why this region is also have to remove from the TI plasmid. Okay, then uh, the transfer of tDNA which begins from the right border doesn't always end at the left border. So rather vector DNA sequence past the left border are often transferred. Okay, so sometimes the left border is also required, but as I earlier told you, the experimental process have seen with the right border. When you delete this right border, then the transfer process will be also inhibited, but it is not with the left border sequences. Okay, so the right if it is started with the right border, then it is not necessarily like it will end with the left border. Okay, so before that also it can be means the procedure can be happen. So this thing also we can remove for the when we are using this TI plasmid as a vector system. Okay, so when this vector system containing the following component, then only it can be used as a cloning vector system. So the first thing that is required is the selectable marker gene. Okay, such as the neomycin phosphotransferase gene we have to add in this plasmid so that it convert the kanamycin resistance on the transformed plant cell. Okay, so which is means those cell which is having this kanamycin resistance gene, then those cell only will grow on the plate which is having the kanamycin resistance. Okay, so suppose this is the agar plate. Okay, so when you transformed your cell with your target gene, okay, so your target gene is having kanamycin resistance. Okay, so this plate agar plate is having kanamycin. Okay, so other transformed cell which is not having this plasmid or which is not having the kanamycin resistance will not grow on this plate. Okay, so that's why one selectable marker gene will required to transform the cell. Okay, so this gene is put under the control of transcriptional regulation signal, including both promoter and termination side. Okay, and another thing is origin of replication you require when you wanted to means clone in the E. coli system. Okay, which is very preferable system in the labs. So we generally do clone in the E. coli system. So that's why, because it is having the origin of replication of agrobacterium tumefaciens, so you have to replace this origin of replication with the E. coli origin of replication. Okay, then only you can use this TI plasmid as a cloning vector. Okay, the third thing is the right bordered sequence. Okay, because it is very much essential for the DNA integration into the plant DNA, plant cell DNA, and most cloning vector include both right as well as the left bordered sequences. Okay, and one multiple cloning site is required, because in the tDNA region there should be one multiple cloning site. So multiple cloning site is what, where your target DNA will be inserted, and multiple cloning site means there should be like different variety of restriction enzyme can be included, and every enzyme should be having the single restriction site in those multiple cloning site. If you will see the ideal plasmid structure, there is a one multiple cloning site. Okay, so multiple cloning site means there is a variety of restriction system. Okay, suppose this is the hind1 hind3 bamh1 eco-r1 like that, and nd1 xho1 like that. Different variety of restriction site will be there from the C terminal as well as the N terminal, and in the middle of this N terminal C terminal there will be your target gene. Okay, so that is the ideal plasmid. Okay, or ideal cloning vector. Okay, and each enzyme should be having single restriction site in the multiple cloning site. Okay, so like that here the multiple cloning sites should be inserted into the this TI plasmid. Okay, to harbor the your target gene. Okay, and the next thing is the killer gene or the encoding or toxic gene downstream from the left border to prevent unwanted vector DNA past the left border from being incorporated into the transgenic plant. If this incorporation occur and the killer gene is present, the transform cell will not survive. Okay, so here also in the after left border sequence in the downstream part there should be one killer gene. Okay, so suppose if your target gene will not integrated and some other gene from other gene of the plant will inserted here, then what will happen is this killer gene will get activated and it will not allowed to survive on the kanamycin plate. Okay, so this killer gene is also, it is optional thing, but the selectable marker is very much essential when you are using this TI plasmid as a vector system. Okay, here the selectable marker gene and one origin of replication, then right border sequence and one multiple cloning site. Okay, this here one I forgot to mention, the very much important thing is the virulence gene is also responsible means virulence gene is also responsible when you are using this plasmid as a vector system, because this virulence gene is having the capacity to transfer this tDNA region from one cell to another cell. Okay, so apart from this all things, you have to also use the virulence genes. Okay, so this is the procedure how the transfer happens. So what happens is when the plant cell is injured, then some phenolic compound will be secreted by the plant cell. Okay, in the site of the infection, then this agrobacterium tumefaciens through the signaling it will come and attach to the plant cell. Okay, and this phenolic compound, which the name of the phenolic compound is the acidosyringone. Okay, this acidosyringone will bind to this virulence A gene. Okay, this virulence A gene, here you can see this is the virulence A gene. It will activate this virulence A gene. Okay, and this virulence A gene is connected with the virulence G gene. Okay, and after binding this phenolic compound to virulence A gene, it will activated this virulence A gene, and it is automatic activated by the autophosphorylation, because it is a kind of kinase. Okay, so when once this phenolic compound is bound to this virulence A gene, then it will activated, and after activation of virulence A gene, it will activated the virulence G gene. Okay, and once this 2 gene will be get activated, this virulence G gene will go to the cytoplasm, and this virulence G gene will be responsible for the activation of other virulence genes. Okay, there are lots of virulence genes. So first in the membrane region there is two virulence gene, one is the virulence gene A and another is the virulence gene G. Okay, so this phenolic compound which is secreted by the infected plant is sensed by the virulence gene A. Okay, once the virulence gene A activated, it will activated the virulence gene G. Okay, and this virulence G will go to the downstream part and it will activated the other virulence gene which is present in the TI plasmid. Okay, so then the second thing will be, what will happen is the second thing is the cut. Okay, so cut means now the tDNA region is present as a double stranded DNA. Okay, so for the transfer of the for the transfer of the tDNA, it should be present as a single stranded form. Okay, so the then through the help of virulence D1 and D2 gene, this tDNA will be converted into the single stranded tDNA. Okay, and once it is get converted into the single stranded tDNA, it is very prone for the nucleus attack. Okay, it is very prone for the nucleus attack. So that's why there is two type of virulence gene. One is the virulence gene E2 and another one is the virulence gene D4. Okay, these two gene will be bind to this single stranded tDNA, and it will protect from the nucleus activity, as well as it is having the signal from for the transfer from one cell to another cell. Okay, so this gene will bind to the single stranded tDNA, and it will help to transfer from one cell to another cell. Okay, and the transfer happen through one channel which is called as the T4 SS system. Okay, and it is this complex is made by the virulence gene B. Okay, these two genes, virulence D4 and virulence B gene, form this complex of the transfer. Okay, this system here you can see that this system is a channel. It is making a kind of channel for the transfer of tDNA from agrobacterium to plant cell. Okay, so once this channel will form, it will transfer from agrobacterium to from the plant cell. Okay, in the plant cell, what will happen is this through the help of right bordered sequence, there will be the non-homologous recombination in the plant genome. Okay, this is the plant genome, suppose. So here the non-homologous recombination will happen, and this non-homologous recombination will be happen due to the presence of right bordered sequence of the tDNA. Okay, and it will integrated into the plant genome, and after integration there will be like if suppose your target gene is present, then here the production of your target protein, and if it is natural condition, then because it is having the cytokine and auxin and opine gene, that's why it is synthesizing the cytokine, auxin and opine. Okay, and it will cause the tumor. And when you are having the target gene, then it will produce the target protein. Okay, and it will create the modified plant. Okay, because it is having the selectable marker. So those cell which is having this target gene, we will select based on the neomycin or kanamycin resistant. Okay, and then we will isolate those cells which is having our target gene, and then we will grow in the preferable media to produce the modified plant. Okay, so this is the complete process of how the TI plasmid transfer from the agrobacterium tumefaciens to plant cell. Okay, so once again we will see, like because this procedure is very much important. So this is the TI plasmid, suppose. So the first thing is attachment. Okay, so how the attachment will happen? If suppose the plant cell, the dicot plant will be infected, then from the infection place there will be release of the acidosyringone, which is a kind of phenolic compound. Okay, this phenolic compound will be sensed by the virulence gene A, which is present on the membrane of the agrobacterium tumefaciens. Okay, here the virulence gene A will be present. Okay, it will activate the virulence gene A, which is a kind of kinase. Okay, there is a virulence gene G which is attached with the virulence gene A. It will activate, once it is activated, it will activated the virulence gene G. Okay, so once the virulence G gene activated, it go to the downstream part. Okay, after releasing from the virulence gene A, it will go to the downstream part, and in the TI plasmid it will activate the remaining virulence gene. Okay, which is present here. Okay, it will activate another virulence gene which is responsible for the tDNA transfer. Okay, so the next thing is you have to you have to make this tDNA as a single stranded DNA. Okay, so the single stranded DNA, how you will make? There is a two enzyme. One is the virulence gene D2, virulence gene D2. Okay, so this virulence gene D2 will break the hydrogen phosphodiester bond of this DNA and make into the single stranded DNA. Okay, So after making single stranded DNA, it is very much prone for the nuclease activity. Okay, so that's why this virulence gene D2 and virulence gene E2 will bind to this single stranded tDNA. Okay, and this virulent gene E2 also act as a single strand binding protein. Okay, so it will bind and help to protect this single stranded DNA from the nuclease activity. Okay, and at the same time, this 2 gene is also having the signaling sequences. Okay, so this will direct the tDNA from Agrobacterium tumefaciens to the plant cell nucleus. Okay, so after that it will direct to the plant cell. Okay, and for means for transfer there is a requirement of a one complex channel. Okay, so this complex channel is made up of virulence gene B. Okay, there is a participation of virulence gene B which prepared this complex system or the channel system for the transfer of the single stranded tDNA. Okay, so as it is with the means bind in the binding form of virulence gene D2 and E2, this tDNA will transform to the plant cell. Okay, in the plant cell after means insert into the nucleus, because it is having the signal peptide for the nucleus transfer, this E2 gene. Okay, so in the nucleus once it goes, it is having the right border sequence, and with the right border sequence it will do the non-homologous recombination with the plant genome, and it will integrated into the plant genome. Okay, and after integration it will produce the cytokine auxine opine gene for the crown gall disease causing, or if you are having the modified tDNA plasmid, then it will be having your target gene. That time it will produce your target protein in the plant cell. Okay, so this is the complete mechanism of the how the TI plasmid transfer from Agrobacterium tumefaciens to plant cell. Okay, so here we can see like there is the virulent genes function you should know. Like many exam point of view, it has been asked like how many numbers is having this virulent gene, like this vir A and vir G is having only one number. Okay, and this vir A gene is a phenolic signal sensor. Okay, and this weed gene G is a inducer for other weed genes. Okay, it activate other weed genes. Okay, and then second is the vir B. Okay, vir B is having 11 in number, 1 to 11. This vir 1 to 11 in number will participate in the formation of this channel or complex. Okay, synthesis and assembly of T pilus. Okay, this is called as a pilus, channel or complex. Okay, whatever you want to say you can say. Okay, so it is responsible to making this channel. Okay, this weed gene, and it is in 11 in number. Okay, then you are having the weed C1. Okay, so weed C1 is generally present in the downstream of sorry here sorry where it should be downstream of left border sequence. Okay, why? Because this C1 is a overdrive sequence, means it will enhance the procedure of or I can say it is helpful in the efficient transfer of the tDNA region from one cell to another cell. Okay, so C1 is a work as a overdrive sequence. Okay, it enhance the tDNA transfer. Okay, then you are having that VD1. Okay, VD1 required for the tDNA processing in vivo and for double standard tDNA burden nicking in vitro. Okay, so this D1 and D2 will participate in the making double standard tDNA into single standard tDNA. Okay, but after converting into the single standard tDNA, this D1 will be removed, only the D2 will be attached to the single standard tDNA. Okay, so this is also 2 in number. Okay, so these three genes in like A, G and C is having 1 in number. This D is having 2 in number, E is also having 2 in number. E will act as a single strand binding protein, okay, and it is also having the signal peptide to transfer from agrobacterium tumefaciens to plant cell nucleus, okay. So this is also 2 in number, okay. Then you are having the vir F, which is which is not having any kind of function in the agrobacterium, but in the plant cell it will like this gene will decide like what type of plant it transfer, and it will interact with the SKPL, which is a kind of transcription factor, and it regulate the cell division of the plant cell, okay. So the next one is the VIRG, which is responsible for the activation of other VIR genes. Then you are having the VIR-H. VIR-H is putative cytochrome P450 enzyme. Okay. So it is also not very much essential for the tDNA transfer. It is responsible for the agrobacterium photosynthesis. Okay. So then you are having the VIR-J gene. It is putative T strand binding protein. Okay. It is also act as a E2 protein. Okay. So it is also bind with the T strand, act as a T strand binding proteins. Okay. So these are the different type of VIR genes, and this is their functions, and this is the number as well. Okay. So these three things you have to remember, like what kind of VIR genes is present in the TI plasmid, how many numbers it is having, and what is the function in the agrobacterium as well as the plants. So in this regard, in previous year gate question, it has asked like in nature agrobacterium tumefaciens mediated infection of plant cell lead to crown gall disease in plant, hairy root disease in plant, transfer of tDNA into plant chromosome, transfer of RI plasmid into the plant cell. What will be the correct combination of the given option? The correct option will be option B, because here it is asking about the agrobacterium tumefaciens. So as we discussed, tumefaciens is responsible for the crown gall disease, and rhizogenes is responsible for the hairy root disease. Okay, so here it is asking about tumefaciens. So the option P as well as the option R will be correct answer. Okay, because the TI plasmid is having the tDNA, which is transfer for one cell to another cell. Okay. Agrobacterium tumefaciens is not having the RI plasmid. RI plasmid is present in the rhizogenes type of bacteria. Okay. So Q will be not the correct answer. Okay, because here it is saying hairy root. So hairy root is caused by the agrobacterium rhizogenes, not by the tumefaciens. Okay, tumefaciens is responsible for crown gall disease. Okay, understood?